Hi everyone, this is Josh again, and for those of you who are new to my channel, welcome. Please be sure to like this video and to subscribe so that I can keep producing awesome content like this. So in this video, we're going to be talking about peptide bond formation, and it's a very important topic if you're taking a class in biology, biochemistry, even chemistry, as well as especially for the MCAT. So the big question is, how do peptide bonds form? Well, peptide bonds, they're made up of one amino acid connected to another amino acid, and they join together in order to form a polymer chain known as a polypeptide. And if a polypeptide gets long enough, it can make a protein. And proteins are everything when it comes to biology. That's the reason why we have DNA and RNA, in order to express proteins and to make a protein product. So how do we form a peptide bond? We need to get into the organic chemistry of it specifically. In order to do that, we need to break down the structure of an amino acid. An amino acid here, it has an amino group, usually to the left, if it's an L amino acid, which is the type of amino acid that exists within our bodies. You can have the amino group be to the right, as in this other diagram that I'm showing here. However, if the amino group is to the right, and the carboxyl group is to the left, that is called a D amino acid, which is typically most commonly found in bacteria. However, for the purposes of this video, I'm going to be going over the more common amino acid, which is the L amino acid. It has the amino group to the left, the carboxyl group to the right, the hydrogen at the top, and the R group at the bottom. And that is the typical structure of an L amino acid. Now, in order for the peptide bond to form, what we need to have is have the amino group of one amino acid attack the carbonyl carbon of the carboxylic acid of the other amino acid. And the way in which it's going to do this is that the amino group of the first amino acid, it's going to donate its lone pair to nucleophilically attack the carbonyl carbon. And the carbonyl carbon is going to end up shifting its electrons up to the top of the oxygen. That oxygen will end up becoming negatively charged in order to make a tetrahedral intermediate. And then this other oxygen is actually going to favor deprotonation of the amine hydrogen from the amine group. So much so that we have now generated water as a leaving group. And water is going to leave. And once it leaves, the other oxygen is going to end up donating its lone pair back to the carbonyl carbon in order to regenerate the carbonyl. And now we have made a peptide bond in which the functional group of the peptide bond specifically is called an amide. It's called an amide because we have a carbonyl directly bonded adjacent to an amine group. That is what is called an amide. Hopefully this quick video ended up helping. Please be sure to keep watching for more in this biochem and chem series that I'm doing right here. Take care.